All right, so what we're going to get into now is looking at taking that inner cell mass, right? So taking that embryoblast and essentially transforming it from being this kind of collection of cells, this kind of large mass of cells, and ultimately changing it into a two-layered disc. Okay, a two-layered disc. So this is this disc that's going to be formed is called the bilaminar embryonic disc. Right. So so again, we're changing our embryoblast into this bilaminar disc. So if we look here, what we can see, and what you can kind of see hopefully in some of these images, is this is occurring around the end of the first week. So day 7, day 8, day 9, somewhere around there. Okay. The specific days, don't worry too much about the specifics of the days when these events are occurring, but think of the big idea. Think of this as like a big story, right? Because this is essentially the story of you, right? This is how you were formed and developed through this process. Okay, so again, don't focus so much on the days, focus more on the big picture, what's actually happening, right? So we're taking that embryoblast. Okay, we're taking that embryoblast, and what's going to happen is there's going to be a kind of dividing or a separation of these two cells. Okay, so if you look off to the right, you'll see kind of a blue set of cells and a yellow set of cells. That blue set of cells is going to be called the epiblast. Okay, epiblast. Epi meaning above or on top. Right. The one below, you can probably guess, that's called the hypoblast layer. Right. So you have an epiblast above, and you have a hypoblast below. The hypoblast is going to be next to that blastocele, right? So that blastocyst cavity, blastocele, all of this space that we see all in here, right? This is all the blastocele. And then the epiblast, there's going to be a differentiation, or this kind of further division of cells above the epiblast and then the epiblast itself. So there's going to be this opening, and that opening is going to be called the amniotic cavity, right? So you've probably heard of the amniotic cavity. It's ultimately what surrounds the fetus, right? And we'll look at how it surrounds the fetus here, and the embryo and the fetus here in just a little bit, right? So we've got this two-layered structure, one layer of epiblast cells in blue, right? One layer of hypoblast cells in yellow, right? So you can see that here, right? So bilaminar embryonic disc, a process that we're going to talk about here in a little bit, called gastrulation, is essentially taking this two-layered disc and transforming it into a three-layered disc. So it's not going to be the trilaminar disc, or it's not going to be the bilaminar, it's going to be the trilaminar, right? But for now, stick with two layers. We have an epiblast and we have a hypoblast, right? Another thing to begin thinking about is the fact that essentially all of our cells in our body, all of our tissues, are ultimately going to be derived from the epiblast. Right, so just keep that in the back of your mind for now. Pretty much all of our tissues are going to be derived from these epiblasts. Not every last one, but a lot of our cell tissues are going to be derived from these epiblast cells. Right. Now, what we can also begin to see happening a little bit later, so starting around day 10 or 11, is the formation of these membranes. Okay, so one of these membranes is going to be called the yolk sac. Okay, the yolk sac. So essentially the yolk sac is formed from the proliferation of the hypoblast. Okay, so remember, if we come back here, we've got these yellow cells. So notice here in this image here we've got a layer of yellow cells. These are the hypoblast. These yellow cells are going to begin proliferating around and line the inside of this blastocele. So remember this inner lumen here, this is called the blastocyst cavity or the blastocele. Those yellow hypoblast cells are going to proliferate and line the inside. Okay. When that happens, that inner space is going to be called the yolk sac. Okay, the yolk sac. If you were a bird or a reptile, it would have yolk in it, right? And that's how you would get your nourishment. Okay. But for us, we are mammals, so we are getting our nourishment through the placenta, right? And again, we'll talk about the placenta here in just a little bit, but it's continuous with that hypoblast layer, right? So those yellow cells are proliferating, they are encircling the blastocyst cavity, and you form the primary yolk sac. Okay. Now you've also got this opening forming again, kind of around the epiblast cells, right? And so you create that opening in there. That's called the amniotic cavity, and the membrane associated with it near that epiblast layer is called the amnion. Okay, it's called the amnion. So we've got our yolk sac, we've got our amnion, again associated with the amniotic cavity. It's going to be fluid filled. It's going to surround the embryo and the fetus ultimately, right? The water, or rather the fluid in the amniotic cavity. That's the water when you've heard of the term, a woman's water breaks, right? That's the amniotic fluid, right? That is in this cavity that's ultimately going to surround the embryo, right? I know at this point it's hard to visualize and conceptualize how that's going to occur, but here in a little bit we're going to talk about folding, and you'll see how that amniotic cavity is ultimately kind of brought around and wrapped around the entire embryo and then ultimately around the entire fetus. Okay, so we've got our yolk sac, we've got our amnion. Another one that we have is called the chorion. Okay, the chorion. So the chorion, if you notice here, all this kind of pink tissue. Okay, all this pink tissue. This is something called extraembryonic mesoderm. Right. We'll talk about what mesoderm is here in just a little bit. But essentially the chorion forms when you take that extraembryonic mesoderm, separate it into two, and it's this outermost layer all out here. So all of this here, this cavity is called the chorionic cavity, and then this layer all out here, kind of lining the very outside of the this entire kind of blastocyst structure, that's the chorion. Okay, so it's formed from this the outermost extraembryonic membrane formed from cytotrophoblast and those cysio-trophoblasts, and part of it is going to give rise to the placenta. Okay, so the placenta, as we'll see, the placenta is partly formed from the fetus, from the fetal line. Part of it's also formed as part of the mother as well, right? So the female. So what's happening here is this placenta is allowing for the exchange of nutrients, waste products, gases, and whatnot between fetal circulation and maternal circulation. Okay, there's also the passage of antibodies that can move between the fetus to confer a little bit of passive immunity as well, right? And so what this structure is, and I want to show you a picture here, is essentially a means by which you can have blood from the maternal circulation and fetal circulation not mix, but come in very close contact with each other, or they come very close to each other, such that you allow for diffusion to occur and the movement of gases, waste products, nutrients, other materials, other substances from one circulation to another. Okay. So importantly, the blood's not mixing. Okay, the blood is not mixing. These large spaces here, these large, what are sometimes called lacunae, this is fetal blood. So this in here is all fetal, and then these down here, these blood vessels are maternal. Okay, so it's not mixing. Okay, the blood is not mixing. If you mixed, you could have a potential issue. We've talked about this in previous lectures with that erythroblastosis fatalis, right? The recognition of a Rh antigen on the fetal blood cells and an Rh negative mother, right? We've talked about that in previous lectures, right? So bringing blood to and from this placenta, in terms of from the fetal perspective, are found in the umbilical cord, and you typically have two arteries and one vein. Okay, so the arteries are taking blood to the placenta. So think waste products going from the fetus to the placenta and then ultimately to maternal circulation and then out. And then the vein, usually a single vein, is taking blood from the uterus, from the placenta to the fetus. Okay, to the fetus. So recall how in previous circulation lectures we've talked about how you define arteries and veins, right? So going to the heart or away. So in this case, the vein is going to the heart. It's going toward the fetus, but it's carrying the nutrient-rich blood that it picked up from the placenta, ultimately from the mother. Okay. So again, you've got a couple of different things here that are making up the placenta. So part of it, again, is from that chorion layer that we mentioned, so that outermost extraembryonic membrane, and then part of it is from the uterus, so from the female, right? So again, it's both. So sometimes this term is called, the term for this is chimeric, meaning it's derived from two different individuals. So it's part of it is from mother, part of it is from fetus. Okay, and then you have this connecting stalk going from the embryo to the placenta that will ultimately turn into the umbilical cord, which contains those arteries and typically the single vein. Okay, so I want to show you just a little bit more detailed picture about the a detailed picture of the placenta, just to show you exactly what it's made of. Okay, so I'm not overly concerned if you understand or if you know all of the specifics of the placenta, but some of these structures that make it up will be, or rather should be, familiar to you. Okay, so if you start here we have our epiblast and hypoblast. So notice here blue and yellow. Here's our amniotic cavity. Here's that yolk sac that's kind of getting pinched off. Ultimately, there you have that chorionic cavity, and then this outer layer is the chorion, right? So notice here we've just zoomed in on a layer of this chorion, essentially, right, which is made up of some of that extraembryonic mesoderm, syncytio, trophoblast, cytotrophoblast. So here you can see those layers: extraembryonic mesoderm, cytotrophoblast, syncytio trophoblast, in kind of this light green color, right? These are called trophoblastic lacuna. These are essentially going to fill with blood. Okay, these are going to fill with blood. But these three layers here, the extraembryonic mesoderm, cytotrophoblast, syncytio trophoblast, they're going to form a villus. So they're going to form this large extension that kind of dives deep into and in between these lacuna, and this is going to form these villi that allow for the exchange of material between between fetal circulation and between maternal circulation. So think oxygen, carbon dioxide, nutrients, waste products, all of those things are going to be moving in specific directions. And think about the direction that those things are going to move based on what they are, right? So if it's oxygen, it's going to be moving from maternal blood to fetal blood. If it's waste products like CO2, it's going to be going from fetal blood to maternal blood. Right? If it's other nutrients that are for the fetus, then obviously those would be moving from maternal circulation to fetal circulation. Okay, again, all without the actual blood mixing. Okay, so that's important. Right? So these structures here are given different names depending on what their actual anatomy is. So primary, secondary, tertiary, you don't need to worry about that, but just know that it's formed from those various layers, and then again meeting up with part of the actual endometrium from the uterus as well. Okay, so these characteristics of the placenta, again, they're allowing for the exchange of material, waste products, gases. It's attached to the inside of the uterus. We'll look at a picture kind of exactly where it's at. It's also selectively permeable, so allowing certain things to pass through, other things to not pass through. In some cases, potentially dangerous things may be able to pass through. The placenta has to be expelled after birth. So you birth the fetus, and then you also have to expel the placenta as well, which is sometimes referred to as afterbirth. Okay. So here are some things that potentially could cross the placenta, resulting in potential, side effects for the fetus, maybe birth defects, things like that. So viruses, bacteria, drugs, alcohol, toxins. So something you've likely heard of is fetal alcohol syndrome, right? And so this wouldn't be a concern as much if alcohol didn't cross the placental barrier, but it does, right? Nicotine, caffeine, right? All these different things. So this is why pregnant females are urged to not smoke, don't drink alcohol. Be careful what medications they're taking. Are those medications teratogenic, meaning can they cause fetal harm, birth defects, things like that? and then other medications as well. And then there's other things that the pregnant woman is urged to take, right? So a really good example of this is folic acid, right? So for the prevention of neural tube defects in the fetus. All of these have to do with that permeability of the placenta to various materials and substances, right? So keep that in mind. So this here is just kind of an overview of these last few things that we've talked about, right? So the formation of some of these membranes. So again, we've got epiblast and hypoblast. So take this blue little layer here as epiblast. Take this yellow layer as the hypoblast. Here's the yolk sac. Here's the amniotic cavity. Here's the chorionic cavity. Here's the chorion, right? That outermost extraembryonic membrane. And then the chorion and this functional layer of the endometrium will form the placenta, right? So here you can see some of those villi starting to form, right? Some of those little extensions beginning to form. You can see some blood collecting in there as well, right? So if we took one of these little villi and we zoomed in on it, it would look something like this. So again, you've got maternal blood, we've got fetal blood, umbilical arteries, typically a single umbilical vein carrying oxygen, carbon dioxide, nutrients, waste products to and from the placenta, and then ultimately to and from the fetus as well. Okay. Now, there are ways in which you can access some of this material. So access the chorion, access the amniotic fluid to do genetic testing, right? So to test for chromosomal abnormalities and amniocentesis, to check for also genetic abnormalities with chorionic villus sampling. But important things to note is that there are certain time restrictions on these. So amniocentesis typically can be formed after the 14th week of pregnancy. CVS, the chorionic villus sampling, this could be done somewhere around 10 weeks of pregnancy. Okay. The chorionic villus sampling can also determine the genetic sex of the fetus as well, along with the various genetic abnormalities as well. Right. So there's certain considerations to make if you're thinking about one of these in terms of what does it provide you, right? What can it tell you, and then when can it be done, right? So it can't be done, 5th week, 6th week, 7th week. You've got to wait for 14th week or so for amniocentesis, 10th week or so for the chorionic villus sampling. Okay, so we're going to stop here. And what we will pick up with in our next part is essentially taking that bilaminar disc with the epiblast and the hypoblast and essentially turning that into a trilaminar disc with 3 layers, and we'll look at the details of that then.